Hello and welcome to today's webinar. My name is Joseph Krieger, and today we have the pleasure of interviewing Dr. Marvin Singh on the role of the gut microbiome in general health and wellness. So, Dr. Singh, thank you very much for taking the time to do your presentation on the biochemistry of the gut microbiome. We look forward to hearing from you.
Thanks, Joe and Boston BioLife. I'm super excited to talk to everybody today about our presentation that has packed full of information. Let's get started. Before we start, I want to quickly introduce myself. My name is Dr. Marvin Singh, and I am an integrative gastroenterologist, and I've become very fascinated with the gut microbiome over the course of my career. And this presentation is one of my favorites to give because it's very easy to understand and very educational. So let's get started. I usually start these conversations by describing what the gut microbiome is, and this slide actually is a nice analogy. It shows that there are so many different components to the gut microbiome, and they all work together as a beautiful ecosystem. It's like a rainforest we have inside of our gastrointestinal tracts. And when everything is growing and living together in harmony, this vast ecosystem can have tons of helpful components to it and be healthy for us, just like this rainforest is healthy for our environment. Let's start by going over what some of the healthy characteristics of a gut are. And we'll be coming back to this illustration quite frequently throughout the presentation. It's a really nice depiction of what the healthy gut looks like. So you can see that in a healthy gut, uh, there's good microbial diversity, there's low levels of pathogens, which are bad bacteria, and toxins. The mucosal layer is nice and thick. The tight junctions between the cells are strong, and there's a low level of inflammation. On the other hand, we have here an illustration of a leaky gut or an inflamed gut, and we can see that there's a big difference between the prior slide and this slide. In this slide, there's low microbial diversity, there's an overabundance of mucus degrading and toxin producing pathogens or bacteria. The mucus layer is degraded and inflamed, and the cells get inflamed, and the tight junctions between these cells become weak. And as a result, you can see in the bottom of the picture that an inflammatory response is initiated. Some of the factors that can drive this situation include antibiotics, excessive alcohol, smoking, stress, lack of sleep, intense exercise, actually even lack of exercise, artificial sweeteners, sugars, high saturated fat diet, and glyphosate or Roundup. And there is plenty of literature in the journals describing how diet can play a role in dysbiosis. When we say dysbiosis, we're referring to an imbalance in the gut microbiome. For example, the Western diet, or otherwise known as the Standard American or SAD diet, is particularly low in fiber and high in carbohydrates and fats. And this is one factor that can lead to severe dysbiosis or an imbalance in the gut microbiome. We also know that high fat diets are associated with inflammation, leaky gut, and higher levels of LPS or lipopolysaccharides, which are the bacterial endotoxins. And this can lead to a low grade systemic inflammation. So, this is a common theme we'll be coming back to. What is LPS? So, that's often the next question people ask. So, LPS, as I mentioned, is lipopolysaccharide or the bacterial endotoxin. This is a nice picture kind of showing what the structure is. And we see that when LPS binds to LBP and it approaches a receptor, it can initiate an inflammatory response in the cell. And this is where the low-grade systemic inflammation comes from. As we mentioned, there are different kinds of fats that we can eat in our diet. And these various types of fats, depending on what kind they are, can actually um, cause an increase or reduction in the amount of LPS. And again, high saturated fat diet could cause the levels of LPS to increase, whereas you can see the more healthy omega-3 type of fats can actually cause a reduction. And the monounsaturated fats like olive oil may actually cause a neutral response, almost like saline itself. High sugar diets are a big thing as far as a contributing factor to imbalance in the microbiome. One of the mantras in my practice is sugar is bad and fiber is good. I keep saying that over and over again, and it's quite true. There's a lot of literature. This is an example of one paper that was published showing how high fat, high sugar diets can induce microbial dysbiosis and gut inflammation. This can also trigger remodeling of the gut-brain axis, which is an important consideration when we're talking about mental health disease specifically. And we all know that high sugar diets can lead to an increase in body fat mass. And a lot of these um, occurrences are due to a modulation of the gut microbiome. So we want to pay attention to that theme as well. And here we are again, back to our illustration showing the healthy gut and the leaky gut side by side. And you can see very easily that there's totally a difference between the two. And we want to stay towards the left pane where we're having a healthy gut, which is in homeostasis. And so we'll talk about how to do that as we go along in the rest of the presentation. We'll start by talking about disruption of the gut microbiome. And so we also have a lot of literature that outlines for us going way back for many years. This is not really a new concept, by the way. We're talking about it more so in more recent times, but a lot of this literature goes back many years, and you can see this is a paper from 2014 which describes how disruption of the microbiome can be associated with obesity, inflammatory bowel disease, IBS, type 2 diabetes, and even colon cancer. So we know that when the microbiome is imbalanced, and we get an increase in chronic inflammation, that this could lead to chronic diseases such as we have mentioned. And when we are able to modulate the microbiome in the other direction, we can actually have very helpful health benefits that can occur as a result. One of the prototypes for conditions that we often talk to or refer back to with regards to the microbiome is inflammatory bowel disease, or IBD. It's because it's a condition where people can have actual inflammation that's visible in the colon. And there's a lot of literature going over some of the microbial changes that occur in the gut in those patients who have inflammatory bowel disease. And some of the themes we notice is that in inflammatory bowel disease, the bacterial diversity is affected. There's a decrease in Firmicutes and an increase in proteobacteria. And there have been trials and ongoing research looking into therapies to correct this bacterial imbalance for the purpose of trying to improve the underlying condition, inflammatory bowel disease. Akkermansia muciniphila is one of my most favorite microbes. It's a very famous microbe. I often lovingly refer to it. It has been associated inversely associated, I should say, with obesity, diabetes, heart disease, and low-grade inflammation. We want a lot of this guy around in our microbiome so that we can have a healthier gut microbiome overall. And we know that having this microbe in our gut can reduce the development of a lot of chronic disease and inflammation. Another one of these good guys, his buddy, Faecalibacterium prausnitzii, has been associated with decreased risk the abundance has been associated with decreased risk in inflammatory bowel disease and can have a protective benefit against the development of inflammatory bowel disease. So this is another really good guy that we want to make sure we have plenty of in our gut microbiome. Next, let's transition to talking about dysfunction of the mucosal barrier. This is another key concept when we're talking about leaky gut, if you recall back to our illustration from the beginning. And so we know that the gut barrier is also an important part of the process, and that dysfunction of the gut barrier is associated with inflammatory bowel disease, autoimmune disease, and systemic inflammation. What happens is that these bad bacteria or these pathogenic bacteria can induce intestinal barrier defects, and then they can translocate to distant sites like the liver and lymph nodes, and this can be a trigger for autoimmune disease. We also see that when the intestinal mucosal layer is dysfunctional and these bacteria translocate, that there's a role in the development of depression. And so we often talk about leaky gut and leaky brain, and leaky gut can lead to leaky brain, which can lead to neurologic conditions and mood disorders like depression, for example. And this is a nice paper that is from way back in 2008, sorry, that goes over how this occurs. Gastroesophageal reflux disease or heartburn is another condition where acid can reflux into the esophagus. And so we know that when this occurs and there's chronic mucosal inflammation, that there's release of interleukin 8, which is a pro-inflammatory mediator from the lining of the esophagus, and that this acid type fluid that has a corrosive effect on the esophageal tissue can degrade the junctional proteins, and this can help contribute to destruction of the epithelial barrier, and as a result, inflammation inside the lining of the tissue. Even in HIV and AIDS, we see how important the dysfunction of the mucosal immune system can be, because in this situation, severe dysfunction can lead to progression to AIDS. And so this also plays a major role in this condition as well. And so this brings us to one of our main topics of discussion today, which is the total gut restoration. We kind of outlined already and showed in the literature how various different factors related to the gut barrier and the microbiome can contribute to the development of chronic disease and inflammation. What we want to do now is go over what some of the solutions are. And so these are the three R's of the total gut restoration plan. And these include recondition, reinforce, and rebuild. And so we'll come back to these three main categories, but just remember the three R's: recondition, reinforce, and rebuild. And we'll start by showing our favorite illustration again, so you can keep this picture in your mind as we talk about the three R's again. And remember that we want to try to push towards the healthy gut, which is on the left side of the screen, and try to heal up the leaky gut, which is on the right side of the screen. And so here we are, the Total Gut Restoration Program: recondition the gut, reinforce the beneficial changes, and rebuild the intestinal mucosa, the three R's. We'll start by going over recondition. So as we outlined, in many chronic diseases, there's low microbial diversity in the gut. So what we want to do is revitalize that garden. We want to remove the weeds, till the soil, and fertilize the plants so that we can have this vast ecosystem that we showed in the very first slide of the presentation. The bacillus spores do this because they can modulate the gut microbiome. They can increase short chain fatty acid production by 40%. And short chain fatty acids are the anti-inflammatory compounds that are made by the microbes in the gut microbiome. And they encourage the growth of these very helpful bacteria that I mentioned earlier that were my favorites: Akkermansia, Faecalibacterium, and also Bifidobacteria. And so Megaspore Biotic is the first 100% spore-based broad-spectrum probiotic that has been shown clinically to improve leaky gut by 60% in just 30 days. It's a unique all-spore formula that effectively reconditions the gut by increasing the diversity and encouraging the growth of key health promoting commensal gut bacteria. And so this is how we recondition the gut. And here's a nice study, I often refer back to it from just a few years ago, showing how the oral spore-based probiotics can be associated with a reduction in bacterial endotoxins. So this is the LPS that we were referring to earlier, triglycerides, and even other disease risk biomarkers. This is a nice study that was done at the University of North Texas on some college kids, and they looked at changes in the serum endotoxin when they got the spores versus the group that did not. And you can see here that there's a quite substantial difference between the two groups. And the spore treatment group had a 45% reduction, whereas the placebo treatment group had a 32% increase. Effectively, that is over 60% difference between these two groups. Our next topic will be reinforce. And so we know the gut microbiome is a dynamic ecosystem that's constantly changing. So we want to build a diverse and healthy garden, but we need the proper fertilizer in order to do that. And this is where the prebiotic comes into play. Oftentimes, many of these prebiotics can feed both the harmful and the beneficial bacteria. So a precision prebiotic is going to be the most helpful because we want to selectively feed the important keystone strains, those good guys we were talking about, Akkermansia, Faecalibacterium, and Bifidobacteria, because these are the ones that are associated with a reduced risk of chronic disease and chronic inflammation. And so this is where Mega Prebiotic comes into play. It's the first precision prebiotic supplement that's made up of clinically tested, non-digestible oligosaccharides that can increase the microbial diversity and selectively feed these good bacteria. It reinforces the beneficial microbial changes that are created by Megaspore Biotic and can promote a strong and diverse microbiome. So this is a key concept and we want to keep coming back to as well. And this is a very nice slide showing that in just a matter of weeks, by giving these oligosaccharides, that we have very substantial increases in populations of these helpful bacteria. So you can see Faecalibacterium, Akkermansia, and Bifidobacteria can increase quite substantially in as short as 1 week's time. And so this is how helpful the prebiotics can be. And then our final R, rebuild, is the next topic. So we know that there's a thick gel-like mucus layer that protects the intestinal lining from damage. And many chronic diseases are linked to the breakdown of the intestinal mucosa, as we kind of outlined in the beginning of the presentation. Butyrate, that short chain fatty acid that's anti-inflammatory, that is helpful, that we referred to earlier, can increase the mucin production by upregulating MUC2 genes. The bacillus spores and 4 key amino acids can rebuild the intestinal barrier by modulation of the tight junction proteins and butyrate producing microbes. And so this is where megamucosa comes into play. Megamucosa is the first complete mucosal support supplement of its kind, and it's formulated with these key amino acids to help rebuild the healthy mucosal barrier. It also contains dairy-free immune globulins that have been clinically shown to support a healthy immune response in the mucosa, and a state-of-the-art flavobiotic, which is clinically shown to support microbial diversity and also alleviate barrier dysfunction, which we're referring to as leaky gut. And so this is a nice article showing, talking a little bit more about these mucus building blocks. L-threonine, L-serine, L-proline, and L-cysteine are these amino acids we were referring to, and they increased colonic mucin synthesis by 95%. That's quite substantial, so you can see how helpful this can be. Polyphenols in the gut microbiome are an important topic as well. We know that dietary polyphenols can modulate the intestinal inflammatory response. They can reduce intestinal inflammation. They can block stress-activated pathways. They can protect against experimental colitis and reduce the risk of inflammatory bowel disease. And citrus polyphenols have an impact on the gut microbiome as well, and you can see a quite substantial increase in butyrate production, reduction in fecal calprotectin, which is a nonspecific marker of inflammation in the gut, and can increase certain Clostridium cluster by 44% after a short 12 weeks. Bovine immune globulin is actually one of my favorite components of a gut healing protocol, and there is quite substantial amount of literature published about how helpful this can be. So we're referring to serum-derived bovine immune globulin, and there is one study here that we're showing how this product can increase intestinal mucosal CD4 lymphocytes and promote intestinal repair in the setting of HIV enteropathy, so it could be quite helpful. In the setting of pediatric ulcerative colitis, there's a case study demonstrating that it can help heal the underlying mucosa. And in the case of C. diff, which is a bad diarrheal infection, it has been demonstrated to bind and neutralize several toxins from C. diff strains. So this can also be very helpful therapy in that setting. So here we are, the total gut restoration, recondition, reinforce, and rebuild with the Megaspore Biotic, Mega Prebiotic, and Mega Mucosa. Now we want to switch gears here real quick and talk about this brand new test called BioMFX. So we know that we need to follow the total gut restoration program in order to help heal the gut. But how do we assess the gut microbiome? And this is an exciting new test that is now available that I want to talk to everybody about. So BioMFX is the most accurate analysis on the market. There's a high DNA capture when you do this stool test. It's a stool test of the whole microbiome. And important to underscore is that it's a functional analysis of the microbiome. And in the report, you can get some customized lifestyle and dietary recommendations. And so this is a very cost-effective way of understanding what's going on in the gut microbiome so that you can understand how to complement your therapy accordingly. We'll go over briefly some of the key parameters that are important and that are reported with this new report. We know that one of the things that everybody agrees on is that diversity is an important component of the gut microbiome. And so we have two diversity scores. The alpha diversity score tells you how diverse your personal microbiome is. And the beta diversity score gives you an idea of how diverse your microbiome is compared to healthy individuals in your population. So you can get an idea of that comparison. Two of my favorite parameters that are evaluated are the resistome and the pathobiome. So we know that in the microbiome, everybody has some bad bacteria in addition to good bacteria. It's not necessarily more concerning that there is a few bad bacteria, but what would be more important to understand is what the net effect of their presence is in the presence of the good bacteria. So how well are the good bacteria actually controlling these pathogenic bacteria? And so this is a score that gives you an idea of that, the pathogen control index. And so it can be very helpful. You know, if you're in the red zone, 0 to 2, you have an idea that there's something bad going on there, as opposed to being towards the green side where you know that you may have some pathogens, but the microbiome is doing a good job of keeping control over them. Another very unique and interesting parameter that is evaluated is this resistome. And the resistome has more to do with gut resilience and richness. And so we know that if you have high diversity, that means that you have a lot of different species of microbes. But we want to know how many quantities of each of these species there are. So that's what we refer to as richness. Because if you have high diversity in your microbiome, but the richness is low, then you can have a setup for dysbiosis or a gut imbalance. And I kind of try to relate this to politics to make it easy to understand. So if you had 5 political parties and they are all equally able to debate each other and have policies and able to present their thoughts and views, then you may have more of a situation of democracy. But if one of these or two of these political parties start to become more dominant, and they do so as a result of the other parties not having very many members in them, then you can imagine how there could start to become some political imbalance or instability. And this is more of a tendency towards perhaps inequality in the political system. And so you can see in that analogy, this is kind of what could happen in the gut microbiome itself. And in this illustration, we see that our comparison of our sample to healthy Americans, we can see that the resistome is off tilt and towards the red zone. So we will want to work at building the richness in the microbiome, and we'll go over some of the indices that will be demonstrated later that will help us improve this score as well. There are some dysbiosis ratios, which are very helpful as well, that are reported in this test. So it gives you an idea of not what each individual species looks like, but what the ratios of these different microbes are like, because that's more important to understand. And when we see where our ratio is compared to the healthy range, and then look at what recommendations can be given dietary wise, lifestyle wise, or supplement wise, then we can help supplement our overall gut healing regimen. Another way of looking at the microbiome is looking at some of the functions of the microbiome. So a good example is with regards to ammonia. If we have microbes that are high ammonia producers and also microbes that are not very good at clearing the ammonia, then we may have a problem in the gut microbiome that may not be favorable. And so, as a result of that, we'll want to know that we should watch how much protein we have in the diet. And we can take certain probiotics that contain Bacillus subtilis, for example, that can help support healthy blood ammonia levels. And so you can see how very helpful looking at functions in this way can be when you're building your gut healing program. And we talked earlier about improving the richness in the gut microbiome and helping to improve that resistome score. And this is one way that we can do that. We can look at the keystone species in the gut microbiome, and we can see, well, how much Akkermansia do we have in the gut microbiome, and how does that compare to a healthy individual? And if I'm low, what can I do to eat in a particular way that can improve that? What type of lifestyle modifications can I make? Are there certain supplements I need to take to help boost the presence of Akkermansia in my gut microbiome? So this is also a very helpful portion of the report. This is another way of looking at the microbial composition in the report as well. So they show what a healthy population looks like on the left, and then they compare that to your sample so you can get a quick idea visually of how close or far off your microbiome is. And they show the percentile level abundance of certain bacterial phylum in your gut microbiome, so you can get an idea of how close to average you are and not, and what particular microbes may be the ones that are most off balance. So in conclusion, this is a functional microbiome analysis that can be very important and helpful in the course of evaluating somebody's gut health and helping them to improve their gut health. It's not a diagnostic test, so we want to underscore that. None of these microbiome sequencing tests are really diagnostic. This should be considered more of a tool to help in the functional analysis of the gut microbiome. And the most important thing to underscore is that we're looking at the whole genome, not just a certain part of the genes or certain particular microbes. We're looking at the whole genome in the microbiomes, and that's very important. And the other important thing to remember is that you're going to get actionable recommendations, because at the end of the day, if you have a report with a bunch of information, it doesn't really mean too much to you if you're not really sure what to do with it. So the actionable recommendations are a key part of the report. And with that, I'll conclude. Thank you.
Dr. Singh, thank you very much for that very concise presentation. I think that the gut microbiome, as many may or may not know, is the gateway to a lot of health issues, especially surrounding inflammatory diseases. And given the recent initiatives with the COVID virus and people looking at immunity in general, I think this is a good time for people to explore not only their personal gut microbiome, but also what they can do to enhance it and change how it functions. And I think that Microbiome Labs has great products for this, and I like the fact that you're coupling it with testing. So, in the role of personalized precision healthcare, this is a very useful tool, especially today, for individuals to take control of their health, to do a better job at building up their immune system, and to be able to have greater resources against the COVID virus, as well as general health and wellness. So, thank you so much for being part of the Boston BioLife webinar series.